Hi everyone, I am Louie Lopez, and currently I am a PhD student at the Academia Sinica in Taiwan. Today I'll be presenting a paper that aims to compare metatranscriptomics with PCR-based methods such as metabarcoding with morphology in estimating the diversity and composition of zooplankton communities. To start, zooplankton communities are complex metazoan communities, which often consist of different species coming from different phyla, and at the same time we encounter different developmental stages coming from the same species. Traditionally, we are using morphology in studying this type of community, but this has some limitations, especially when you're dealing with thousands of individuals, and at the same time, it would require high taxonomic familiarity to determine which species is this one and which species is the other one. With these limitations, we're now slowly transitioning with molecular-based methods that uses DNA, for example, metabarcoding, and lately, there are some emergence of RNA-based analysis that allows them to have an overview of the functional analysis of the entire community, aside from the usual diversity analysis that we're doing using DNA-based. Advantages of molecular methods are quite apparent. It's rapid and efficient. It also provides higher resolution data, especially with the availability of next generation sequencing. At the same time, it helps us to avoid the taxonomic requirement when you're dealing with the morphological based analysis, especially with the identification of nuclei or copepodites when it comes to copepods or other different developmental stages per species. And then lastly, it enables us to have an overview of the functional analysis, especially with the use of RNA nowadays. But despite these advantages, there are still some methodological uncertainties when it comes to using molecular tools. For example, the use of genomic DNA involves different biases. For example, we have pseudogene contamination that can lead to inflation in terms of diversity estimation. So the genes are copies of, for example, mitochondrial genes that are inserted in the nuclear genome through time and it tends to accumulate. And when you try to amplify a certain marker or mitochondrial marker, you tend to amplify both copies from the mitochondria and the nuclear genome. That's why it's becoming inflated. Another one is that since it's dependent on PCR, it involves PCR amplification bias and the issues when it comes to the universality of the primers being used for analysis of the community. But in order to avoid pseudogene contamination, some literature basically used RNA-based method. For example, they synthesized complementary DNA from the mRNA transcript because it's known that pseudogenes, or most of them, are not functional and not being transcribed into mRNA. However, this approach is still dependent on PCR. That's why the PCR amplification bias and the primer issues are still there. And then lastly, there are some emergence of mRNA-based analysis or our mRNA transcript that avoids pseudogene contamination. And at the same time, it's not dependent on PCR because you're basically extracting the sequences, for example, CO1 sequences bioinformatically. However, this type of method still has its own inherent biases or uncertainties, such as RNA degradation or effective the use of effective transcript assembly tools for the downstream analysis. Given these methods, I basically classify them into two, which are PCR-based methods that includes gDNA and cDNA, and on the other hand, we have metatranscriptomics that uses mRNA transcript. The primary question that we want to address is that which molecular-based methods can help us avoid as many biases as possible. Would it be the PCR-based methods or the transcriptomics? That's why the primary objective of this study is to compare these methods together with morphology as the standard approach for studying zooplankton communities. And once we find a certain approach that is less biased, we tend to examine it and use it for field samples in terms of its replicability and temporal monitoring in the changes of the zooplankton communities. For the methodology, we basically collect zooplankton from the field and then preserve it in RNA later. Once in the lab, we sort it individually It's basically dominated by cladocerans and copepods since we're using freshwater zooplankton. And then we constructed the mock community, and then from the mock communities, we extract genomic DNA and total RNA. And then this is followed by the CO1 amplification using a universal primer coming from gDNA and cDNA synthesized from the total RNA. And then at the same time, we isolate mRNA from the total RNA. And then these three were prepared for library preparation and then undergo sequencing using MiSeq 300 pair end. For the amplicon processing, we use data for the chimera removal, and then the ASV's output of data too is clustered using research so that we have the OTUs, and then Lulu is for the curation of the OTUs. For the transcript processing, we first assemble the transcript so that we can have the CO1 transcript reference. And once we have the reference for each species, we use it for mapping back the raw reads of the transcript, and then we calculate the abundance based on the read level and normalizing the read level using the RSUM. And when it comes to the mock communities we constructed for this study, we have five. It's basically cladoceran dominated, copepod dominated, equal biomass, natural assembly that mimics decomposition in the field, and then with a rare species. We noted the number of individuals placed in each community together with their corresponding biomass. And then in terms of the result, basically we observe inflated OTU number or species richness in the gDNA as compared to the other molecular based methods. So we have a total of 25 in exclusive OTUs detected by gDNA. This inflation in OTUs can be attributed to the co-detection of environmental DNA and at the same time the content of the gut of the samples being used for the construction of the mock community. And in order to have a clear overview of the actual mock community, we tried to filter the actual species that we used, but as you can notice, all methods were able to detect 6 species except for this one wherein it's exclusively detected by the morphology and metatranscriptomics. Both PCR-based method were not able to detect this particular species, and when we observe the primer and the CO1 sequence of the species, there's a primer mismatch. So we're having an issue when it comes to the universality of the primer used in this study. And then going forward, for the detection of the pseudogenes, we basically use the 6 species detected by both gDNA and cDNA. And the expectation is that cDNA doesn't contain the pseudogenes because it came from the mRNA transcript. And the gDNA would contain those pseudogenes. That's why it's expected that the ASV, the nucleotide diversity, and the number of indels would be higher as compared to its cDNA counterpart. At the same time, presence of pseudogenes would make synonymous substitution higher than the non-synonymous substitution. But this particular condition was only observed in the Mesocyclops leucarti, which is a copepod species. So basically, this data doesn't provide us a direct evidence that pseudogenes are present, but we can use this as a preliminary basis for the possible presence of pseudogene contamination in gDNA. And then when it comes to the comparison of the diversity indices in each community using the 4 methods, we didn't observe any significant difference. But however, when it comes to the community assembly presented by each method, basically the gDNA and the cDNA clustered together, while the morphology and metatranscriptomic provides a highly resembled community assembly. So given this data, we can say that metatranscriptomic may help us avoid some of the biases that we experience with the use of PCR-based methods, whether you're using gDNA or cDNA as template. But the next question is that would it provide a consistent data in the field? That's why we tried to collect zooplankton samples coming from a freshwater reservoir starting from July to December 2019. And then we tried to test the replicability and the temporal monitoring capacity of metatranscriptomics in using in field samples. And when it comes to the replicability, Both biological and technical replicates showed highly similar community assembly of the samples. The biological replicates represent different plankton whole from the field, while technical replicates are three different aliquots coming from one biological replicate. And when it comes to the monitoring of the zooplankton communities from July to December, we try to use different markers or such as 16S transcript, CO1 transcript, and site B transcript. And all of these markers basically showed highly similar or highly resembled community assembly per, per sampling date starting from July to December. And wrapping it up all together, what we can learn from this one is that when you're using gDNA, there's a higher possibility that there will be co-detection of non-target taxa or even the co-amplification of the environmental DNA. That's why it's very important for us to have proper preparation of samples. Next, PCR inherent biases, especially the universality of the primers, would be a very important consideration when designing molecular-based approach in studying zooplankton communities, in particular in marine because it's highly diverse and coming from different groups or different taxonomic groups. And then lastly, pseudogene contamination must be considered when you're designing the downstream analysis, especially when you're using genomic DNA as template for your community analysis. And I think the most important question at the end of this presentation is that can RNA-based method, specifically the metatranscriptomics, can be an optimized approach for monitoring aquatic communities? Well, based on this data, it's possible, but I think there's still a lot of things to do, especially to the when it comes to the downstream analysis of this particular approach. And lastly, I would like to acknowledge the members of our team, and should you have questions, feel free to email me or check my information. That's all. Thank you.